Today, we are going to study in vitro haploid production in plants, which is a major technological advancement in plant biotechnology and modern plant breeding. This topic integrates concepts from cytogenetics, tissue culture, and crop improvement strategies. Understanding haploid production is essential, but it enables rapid development of genetically uniform plant lines. In this lecture, we will explore the biological basis of haploid production, laboratory techniques used to produce haploids, and their applications in agriculture. We will also examine the role of chromosome manipulation and hybridization methods in generating haploid plants for breeding purpose. By the end of this lecture, you should be able to clearly define haploids and double haploids and explain why they were important in plant breeding. You will understand how androgenesis and gynogenesis function as alternative developmental pathways that allow embryo formation without fertilization. You should also be able to describe the practical procedures involved in anther culture and microspore culture, explain chromosome doubling mechanism, and understand chromosome elimination in cereal crops. Overall, the aim is to connect basic biological principles with real breeding application. A haploid plant contains only one complete set of chromosomes, represented as N. Normally, higher plants are diploid, containing two sets of chromosomes inherited from both parents. Haploids arise when gametic cells develop into embryos without undergoing fertilization. Haploids are important because they express all genetic traits directly, including recessive ones. Although naturally rare, haploids can be produced artificially through tissue culture techniques. This ability makes haploid a powerful tool in genetic analysis and plant improvement. Haploids versus diploids. Haploid plant contains a single set of chromosomes and therefore carry only one allele for each gene. As a result, all genetic traits are expressed directly, making haploids very useful for genetic studies. However, due to absence of homologous chromosome pairs, meiosis cannot occur normally and haploid are generally sterile. Diploid plants contain two sets of chromosomes and are typically fertile because proper chromosome pairing occurs during meiosis. While diploids are genetically more stable, haploids are extremely valuable for research and breeding because they reveal hidden genetic variation immediately. Importance of haploid production. Haploid production significantly reduces the time required to develop pure breeding lines. Traditionally, breeding method required repeated self-pollination over many generations, but haploid technology allows complete homozygoity to be achieved within one generation after chromosome doubling. Haploids are also useful for mutation breeding, gene mapping, genome sequencing, and hybrid cell production because they simplify genetic analysis and accelerate breeding programs. Haploid technologies are widely used in modern crop improvement. Doubled haploids. Since haploid plants are usually sterile, their chromosomes must be doubled to restore fertility. When chromosome doubling occurs, the haploid genome is duplicated, producing a diploid plant with identical chromosome sets. Such plants are known as doubled haploids. Doubled haploids are completely homozygous at all genetic loci, making them ideal for plant breeding programs. They are widely used in crops such as wheat, barley, and rice because they provide genetically stable and uniform line quickly. Methods of in vitro haploid production. There are three major approaches to produce haploid plants in vitro. The first is androgenesis, where haploids are derived from male gametes such as pollen or microspores. The second is gynogenesis, where haploids develop from female gametophytic tissues. The third method is chromosome elimination, usually achieved through wide hybridization between different species. Each method relies on different biological mechanisms but ultimately results in the production of plants with a single chromosome set. Androgenesis definition. Androgenesis refers to the development of haploid plants from male gametes, particularly microspores or pollen grains. Under normal conditions, these cells develop into sperm cells for fertilization. However, under controlled laboratory conditions, they can be induced to form embryos. This process involves reprogramming the developmental pathway of the microspore. Instead of forming gametes, it begins developing like a zygote and develops into a haploid plant, demonstrating the principle of cellular totipotency. Biological basis of androgenesis. Normally, microspores develop into matured pollen grains that participate in fertilization. However, when exposed to stress conditions such as temperature shock or specific culture environments, the developmental program changes. This reprogramming causes the microspore to follow a pathway instead of gametophytic one. As a result, embryos from direct from pollen cells, which later develop into haploid plants. Androgenesis can occur through two pathways. In direct androgenesis, embryos form directly from microspores without any intermediate stage. This is generally more efficient and produces uniform embryos. Indirect organogenesis. Microspores first form an undifferentiated callus mass. Later, this callus differentiated into embryos and regenerates into plants. Although slower, this pathway may occur in species where direct embryogenesis is less common. General steps in androgenesis. The process begins with selection of suitable flower buds containing microspores. At the correct developmental stage, the plant material is sterilized and cultured under controlled laboratory conditions. Embryos developed from microspores are later regenerated into whole plants. If fertile plants are required, chromosome doubling is performed to convert haploid plants into double haploids. Anther culture involves culturing the entire anther containing numerous microspores. Under suitable conditions, these microspores divide and develop into embryos within the medium culture. This method is widely used because it is relatively simple and allows simultaneously culture of many microspores. It is one of the most common techniques for inducing androgenesis. Steps in anther culture. Unopened flower buds are selected to ensure microspores are at the correct stage of development. The anthers are carefully removed and cultured on nutrient medium under sterile conditions. Embryos or callus develop within the medium and are later transferred for regeneration into complete plants. Proper environmental condition is essential throughout the process. Culture medium requirements. Successful tissue culture requires a well-balanced nutrient medium. MS medium is commonly used because it contains essential mineral nutrients required for plant growth. Sucrose provides energy, while growth regulators such as auxin and cytokines control cell division and differentiation. Vitamins and amino acids support metabolic pathway and enhance embryo development. Advantages of Anther culture. Anther culture is simply to perform and requires minimal specialized equipment. It allows large-scale production of haploid plants from many microspores simultaneously. This method is applicable to a wide range of plant species and is widely used in breeding programs for rapid development of pure lines. Limitations of anther culture. Sometimes somatic cells from the anther wall may divide and produce diploid plants instead of haploids. This reduces the efficiency of haploid production. The response also depends strongly on plant genotype, and some species show very low androgenetic potential. Therefore, optimization of culture conditions is essential. Microspore culture definition. Microspore culture involves isolating individual pollen grains and culturing them on directly. This ensures that embryos originate only from gametic cells because somatic tissues are excluded. This method produces true haploids with greater accuracy. It is considered more precise than anther culture. Procedure of microspore culture. Microspores are isolated mechanically from flower buds and purified. They are cultured in liquid medium under controlled condition. Embryos develop from individual microspores and are later regenerated into plants. The entire process requires strict control of developmental stage and culture environment. Advantages of microspore culture. Microspore culture provides high efficiency and eliminates contamination from somatic tissues. It produces uniform haploid populations. This technique is especially useful for genetic studies and breeding programs requiring precise control over haploid origin. Limitations of microspore culture. The technique is technically demanding and requires precise handling of microspores. Even small deviations in developmental stage may reduce success. Additionally, response varies among species, and not all plants respond well to microspore culture. Factors affecting androgenesis. Biological factor. The success of androgenesis depends heavily on plant genotype and physiological condition. Healthy donor plants produce better responses. The developmental stage of microspores is also critical. Only specific stages are competent for embryogenetic reprogramming. Physical and chemical factors, temperature treatments, nutrient composition, and growth regulators strongly influence androgenesis. Controlled stress is often necessary to induce embryogenesis. Life conditions and culture duration also affect success. Optimizing these parameters improves haploid yield. Ploidy level haploid plants generally lack fertility due to absence of homologous chromosome pairing. Therefore, chromosome doubling is necessary for reproduction. Once chromosomes are doubled, plants become diploid and capable of normal meiosis and seed production. Chromosome doubling method. Chromosome doubling is commonly induced using chemicals such as these chemicals disrupt spindle fiber formation during mitosis. As a result, chromosomes fail to separate and remain in the same nucleus, efficiently doubling the chromosome numbering. Diploidization converts haploid plants into fertile diploid plants. This step is essential for practical breeding applications. The resulting plants are completely homozygous and genetically stable, making them ideal for breeding material. Gynogenesis is the development of haploid plants from female gametophytic tissue such as ovules or embryo sacs without fertilization. It is used when androgenesis is ineffective or when maternal genetic lines must be preserved. Importance of gynogenesis. Gynogenesis is particularly useful in species where anther culture does not work efficiently. It produces maternal haploids. This method is important in crops like onion, sugar, wheat, and sunflower. Methods of gynogenesis. Gynogenetic haploids can produce through ovary culture, ovules culture, or embryo sac culture. Each method involves culturing female reproductive structures under controlled conditions to induce embryo development. Steps in ovule culture. Immature ovules are collected and sterilized before being cultured on nutrient medium. Embryos develop and regenerate into plants used controlled conditions. Factors affecting gynogenesis. Successes depend on genotype, developmental stage, and hormonal balance. Environmental conditions also play a role. Proper nutrient supply and temperature treatment are essential for embryo development. Chromosome elimination technique. Haploid can be produced through wide hybridization between different species. After fertilization, one parent's chromosomes are eliminated. The remaining genome forms a haploid embryo. Example: wheat maize system. In wheat maize crosses, maize chromosomes are selectively eliminated after fertilization. This produces haploid wheat embryos which are later cultured and regenerated. Mechanism of chromosome elimination. After hybridization, one genome fails to divide normally and is gradually lost during cell division. This results in embryos containing chromosomes from only one parent. Advantages in serial breeding. Chromosome elimination is highly efficient and independent of genotype limitations. It is widely used for rapid development of pure lines in cereals. Applications of haploid technology. Haploid technology accelerates plant breeding and hybrid development. It supports mutation breeding and genetic transformation. It also plays an important role in genome mapping and stress tolerance studies. Summary: haploids contain a single chromosome set and can be produced through androgenesis, gynogenesis, or chromosome elimination. Chromosome doubling produces fertile double haploids that are widely used in modern plant breeding. For examination purpose, you should clearly understand the difference between anther culture and microspore culture, as well as direct and indirect androgenesis. These comparisons are frequently asked in conceptual and short answer questions. You should also remember the role of dash in chromosome doubling, major factors affecting gynogenesis, and what wheat cross maize chromosome elimination system, as these are the important applications based topics. To prepare efficiently for CUET PG Plant Biotechnology, consistent concept clarity and regular practice are essential. Structured learning through lectures, notes, and mock tests helps reinforce understanding and exam readiness. Make use of guided learning resources, revision materials, and doubt clearing sessions to strengthen preparation. Continuous practice and conceptual understanding together ensure better performance in competitive exams. Subscribe BioBrain for free lecture series. Subscribe to BioBrains for high quality exam oriented CUET PG Plan by Technology preparation.